Today we're going to talk about epigenetics, which is heritable changes in gene expression, which means that you are the steward of your genome. You are taking care of which genes are being expressed or used by your body right now for future generations. An example of this is twins. They are not the same person, but their DNA is the same if they are identical twins. You guys are probably familiar with Mary Kate and Ashley. One of them actually had an eating disorder and one of them did not, which may create differences in which genes are being used by their bodies. On the left, there is a guy from my hometown who actually was recently killed outside a bar. He was a teacher, but his identical twin brother was a member of a gang, and so they mistook him for his identical twin brother. It was a case of a real-life evil twin. And their gene expression may have been very different because obviously they had been living pretty different lifestyles. On the right, you have a famous actress from Baywatch. She's an actress, and her identical twin sister was a firefighter, and she married a man, and her identical twin married a woman. So there can be a lot of these differences, and some of these differences may be based on genetic differences, but they're not genetic differences in whether or not you have the gene or not, but whether or not you are using that gene or not. And something you can think about is you have a lot of different parts of your body, like you have a brain and you have a liver and you have a finger muscle and you have your eyes, and all of those structures are very different, and you have the genes to make all of those different structures in every cell in your body, but the cells that are in those different parts of your body are using different subsets of those genes, because of course your eyes and your toes are not doing the same thing, and they have to use different genes, although they both have all of those genes from your whole body present in each of those places. So I just wanted to remind you guys a little bit of the DNA structure, because that will be important to understanding how these epigenetic changes actually occur. So DNA, of course, stands for deoxyribonucleic acid, and DNA has two strands. DNA is made of deoxyribose and a phosphate backbone and nitrogenous bases. There are 4 bases: cytosine, guanine, adenine, and thymine. And adenine always pairs with thymine, and cytosine always pairs with guanine. Cytosine will be very important to epigenetic modifications. Here you can see how they actually pair. You can see the molecules that the bonds are between. There are 2 types of nucleotides, pyrimidines and purines. Adenine and guanine are purines, while cytosine and thymine are pyrimidines. And it can be helpful to think of these 2 categories of structure, because in all cases you have a bond between a molecule from each type of structure. So you can see that the bonds between thymine and adenine are similar to the types of bonds that are between cytosine and guanine. When you have DNA methylation, you have a methyl group attaching to the other side of the nucleotide. So you have one part of the nucleotide attached to the other nucleotide, and on the other side from that, basically sticking out of the sides of the DNA, you have a methyl group. And this blocks the transcription of genes, and this can actually be passed on to the offspring. And we'll watch a video now that shows how transcription factors work to give you guys a better idea of what blocking the transcription of the gene would actually look like.
By controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA protein complex called chromatin, In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
The other method of epigenetic change, which is not heritable, DNA methylation can be passed on to your offspring, but this next method cannot be, is histone acetylation, and this is related to DNA storage. The DNA from one cell is actually 3 meters long and coiled, which is about the length of this car, and it's actually pretty fabulous that it is able to be stored inside each and every cell of your body. This is possible because DNA is twisted around histones, which modify how often genes get copied, because histones can wind the DNA more tightly or more loosely. So this can change the expression pattern in you, but it is not passed on to your offspring. And we'll watch a YouTube video to show how that works.
Genetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones, and therefore a condensed or closed chromatin structure.
All of us have about 30,000 genes in our cells. All of our cells are nuclei, so all the cells except for red blood cells have all the genes, but of course they do not express every single gene. If you think about it, different parts of your body are actually completing very different tasks. So think about something like a muscle cell, and then think about the cells in your eye. So very different types of activities are needed. If you think about your eye has a lot of those light and color receptors that the other parts of your body don't need to use, but they all have the genes necessary to produce those types of proteins. Another example that you guys are probably familiar with but haven't thought about in terms of epigenetics is X inactivation, which is exactly what it sounds like. Some of you in this room are male, some of you in this room are female. Those of you who are female have 2 X chromosomes in all of your cells, but at any given time only one is active, because if both were active, you'd have an overproduction of certain types of proteins. And the reason that one X is inactive is because it's coded in methylation and the histones are modified. So the DNA is wound very tightly, causing it not to be expressed, and the methyl groups cause transcription factors not to attach to those genes. So you'll have only one X active at any time. So everyone in this room has only one X active, but whether that's because you only have one X in the case of males or because it's actually being changed by epigenetic factors to not be expressed in the case of females. We talked earlier about human twins. These are mice twins, and the big yellow mouse there and the little brown mouse are actually identical twins. They have identical DNA. The difference is that the mouse on the left has no methylation. Of course, we look at this and immediately we think that, being the big yellow mouse would obviously be worse. But of course, the value of a phenotype depends on the environment that you live in. So in this particular case, a lot of these changes are due to leptin expression, and this expression of this gene is dependent on the maternal care. So if the mother licks the rat a lot, then it changes that gene's expression, and you can see how either of those phenotypes could be valuable. So if you are a little brown mouse that's been cared for by your mama, you don't necessarily need to overeat every time you see food because you've had a reliable food environment experience. If your mama abandons you and you don't get a lot of care from her, or you don't get a lot of care from her because you're in an unstable environment, eating everything you see may be adaptive as well, because if you don't see food very often, you actually won't wind up being overweight. And it's only in the environment where you don't have methylation and you're provided food regularly by crazy scientists that phenotype will show up as so obviously bad. We talked before about how cancer is a disease of mitosis. It's basically where your cells go crazy dividing and the normal mechanisms that serve to regulate cell function don't work. We talked about how you can have problems with cell division and DNA replication such that parts of the DNA get deleted that were necessary for making proteins that coded for, making proteins that regulated the cell cycle. The same thing can happen in the case of epigenetics. You may not lose a gene that was important in making the protein that regulated the cell cycle, but instead you can have that gene be blocked either by histone modification, which will make it not completely inactive, but just not copied as much, or more likely in the case of DNA methylation, where you're actually blocking transcription factors from attaching to that gene and copying it and making the proteins that you needed to regulate your cell cycle. So it doesn't make you a queen in the sense of you as a human being, but there are honeybees, and as we all know, honeybees are not very egalitarian. There's one queen and a whole bunch of workers. Of course, unlike in human societies where people might want to be the queen, in bee societies, the queen basically just is a really fat bee that produces offspring all the time. So not so much fun, perhaps, as being a human queen, but whether you become a worker or a queen, whichever one sounds better to you, is determined by royal jelly, which is a substance that you might be familiar with from some lotions, and it's a substance produced by the bees, and they rub it on certain of the eggs and not on others, and the eggs that get the royal jelly turn into queens, and the other ones turn into workers, and that's because although both the workers and the queens have the genes necessary to become queens, royal jelly actually causes the activation and deactivation of different genes, changes in a pattern of methylation. Today, you guys are going to make your own gene epigenome. So you'll be making it out of pipe cleaners. So you'll have 2 pipe cleaners that represent your DNA, and you'll have different color genes. As you can see here, there are different types of histones, and some of the genes have these yellow things sticking out of the side. So the yellow things sticking out of the side represent DNA methylation, so that is blocking the transcription factor from coming in and copying the gene, and the difference in size of histone represents whether the histone is acetylized or not, so how tightly the DNA is being wound around the storage proteins. Your DNA will begin as 2 pipe cleaners. You'll color in your different genes, and you'll interlace the pipe cleaners into a double helix. Add your histones. If they are not acetylized, they'll all be small. You may need to twist them a little bit so that they will fit more snugly. And then finally, as your persona progresses, you'll be adding different types of histones and methylating your DNA. So this will be based on what your mama does, and your mama in this hypothetical scenario, and what you do. Of course, these are also based on what your mama really did and what you really do, but we can't change that over this course of the next few days, but you can think about this for the future. So in this case, you have some genes. You have a gene for leptin production, a gene for tumor suppression, a gene for insulin sensitivity, a gene for stress response, a gene for muscle fiber growth, a gene for long-term memory formation, a gene for metabolism, and a gene for depression. Of course, in real life, you have 30,000 genes, but just for simplicity's sake, these are the genes we're using here. Leptin production we talked about a little bit with twin mice. Leptin regulates hunger and appetite. Your production of leptin can be changed by your mother's diet. It causes obesity, and if the gene is methylated, overeating is reduced, so you will have less leptin because that gene is being blocked. And this is one of the proteins that was overexpressed in the fat mouse. Then you have a gene for tumor suppression. It's impacted by maternal smoking. It's also impacted by you smoking, and if the gene is methylated, there's less regulation of tumors, and therefore an increased risk of cancer. Insulin sensitivity is the next gene. You are familiar with insulin. You're familiar with it because you've learned about diabetes before, and it's impacted by maternal fitness or exercise, and it's impacted by your exercise, particularly resistance exercise like weightlifting and stuff like this. If your DNA is methylated, you have an increased risk of diabetes. Then you have the gene for stress response that's impacted by maternal stress, and if it's unmethylated, you have more stress hormones produced. Does anyone remember those? The clue is muffin top, and the hormone that we're talking about is cortisol. We've talked about it a lot before. Unmethylation is increased by maternal affection. And what's interesting is actually this photograph that I have here of the pregnant lady freaking out was taken from a BBC article that actually was about a different type of maternal impact. So talking about the impact of maternal stress on asthma. So another type of epigenetic change that we're not going to address in this particular set of genes that we're talking about, but just to give you guys a clue, there are a lot more types of epigenetic changes out there that people study besides what we're going to get to in this lesson. So you can look into that yourselves. Then we have muscle fiber growth, which is impacted by exercise, and we have histone modifications activating genes responsible for muscle fiber production, and we have long-term memory formation, which is impacted by training. In other words, do your homework, and histone modification increases memory formation. We have the gene for metabolism, which is impacted by your parents' diet, and impacted by your diet, specifically the amount of calories consumed. It causes obesity, increased risk of diabetes, and the methylation of genes increases your risk. We have depression, which is impacted by aggressive interactions and abuse, and a histone modification increases depressive behavior. In summary, epigenetics is a change in DNA expression based on your activities, your parents' activities, and your grandparents' activities, and it's caused by changes in DNA's transcription because of transcription factor blocking. And this means that you are the steward of your genome. What you do to your gene expression will impact your health outcomes, but it will also impact the health outcomes of your offspring. And so you are responsible for keeping your DNA expression in good shape for your future generations. After doing the activity, you guys had some questions, and I will here provide some answers. You guys want us to know who discovered epigenetics, and this was C.H. Waddington in 1942. You can tell he's an old-timey scientist because he has a pipe in his hand, and if he understood epigenetics, he would not be smoking like that. But at that time, they didn't fully understand it. He was interested in epigenetics because of what we talked about at the beginning of the lecture on epigenetics, which was that you have a whole bunch of different types of tissues in your body, like your eyes and your liver, and those do different things. And so that was where his initial coining of the term epigenetics came from. He had noticed this phenomenon. Just to remind you guys, there is DNA methylation and histone modification. DNA methylation is where you have methylated DNA, so these little methyl groups on the outside of your DNA attached to your nucleotides that blocks transcription factors from attaching to your DNA and copying that gene. Then you have histone modification, which is a change in the configuration of the protein around which your DNA is wrapped for storage, and that changes how available the gene is for copying. Somebody wanted to know how DNA methylation is inherited because, of course, when you have the two parent strands of DNA and you get the two daughter strands, only the parent strands are methylated, but you have a enzyme that goes around and checks for unpaired methylation and will then methylate the daughter strands of DNA. People are interested in knowing how long it takes twin genes to change because we talked about how twins, of course, will have nearly identical epigenetic changes when they are young, although their genes will be actually identical. Here you can see the epigenetic markers for a 3-year-old and for a 50-year-old, and you can see that the 3-year-old twins have much more similar epigenetic changes, and that makes sense. They've been living in the same environment, presumably, in their parents' house, and the 50-year-old twins have had more divergent lives, and so you can see that their gene expression has changed quite a bit. Can epigenetics cure diseases? Well, there hopefully is a lot more potential for this in the future, but currently there is one drug on the market that cures cutaneous T-cell lymphoma, which is a type of cancer, and it's treated by vorinostat, and that goes in and deacylates histones. We talked about how cancer can be a product of epigenetic changes because, like we talked about also when we talked about problems with mitosis and replication of cells, if you don't have the proteins being expressed that are important in regulating the cell cycle, so you don't have those genes functioning either because you've lost them in problems with mitosis or because you have stopped expressing them because of epigenetic changes, that can be problematic. And so in this case, you can have epigenetic changes caused by these drugs that make certain genes that are important in regulating the cell cycle more available, or others that are causing cancer problems to be more repressed, and hopefully there'll be a lot more opportunities like this to cure diseases in the future. All right, thank you, and I hope that you now understand what it means to be a steward of your genome and that you'll behave responsibly in the future.